training was in human population genetics, and I started working with DNA polymorphisms in the early 1980s to study humans for a variety of reasons, and became involved in the late 80s and early 90s when DNA started being introduced into court as an expert witness. And I testified in many different places around the US and in Canada, and found it quite interesting. It soon became unnecessary for expert witnesses, so I basically dropped out of forensics. It was never my main area of research. And then at the time of the World Trade Center attacks, an expert committee was set up, and I was asked to be on it. It became obvious that the standard forensic markers for individual identification did not work well in a sizable number of cases because of the extreme degradation of the DNA, and because they could say nothing about ancestry of the sample, which would in itself be a help in identifying the person. At that point, I realized that we had all the expertise and the samples necessary to do a lot of research. So I got back involved. And let me then start the lecture and talk about some of what we're doing and why DNA can be very important, both in ancestry and phenotype. And I could say that DNA is going to make everything you've learned so far unnecessary. But I won't go that far. DNA is not going to solve all problems, but it already can be extremely helpful when available, and it's going to be much more helpful in a few years because of projects that are ongoing now. Caveat, what we know now, as I hope you will learn, is often overinterpreted, that there is the implicit thing that if a conclusion is based on DNA, it's immutable, it's true, it's precise. Ain't so. So let's talk about the DNA in the human genome. We've got mitochondrial DNA, which is a very small part of the genome, less than 17,000 base pairs as a circle, compared to the nuclear DNA, which is over 3.3 billion base pairs of DNA. The autosomes are 22 chromosomal pairs of varying sizes. The sex chromosomes, one pair, unmatched. Females have two X's, males have an X and a Y. The Y chromosome can be subdivided into a small part that recombines with the X chromosome at the tip of the chromosome. That is important in segregation during meiosis, forming gametes, and then the non-recombining part, which is inherited without any recombination. So, two parts, they're both interesting and have different implications for how they're studied. All of these segments of DNA have polymorphisms. So what's a polymorphism? it literally translates to a part of the DNA that occurs in many forms. Depending upon how big the segment you look at, one base pair will generally occur only in 2 forms, sometimes 3 and 4. But in general, the least common form must, or the least common form less than 1%, or the most common form at least less than 99%. So we try to make a distinction between a polymorphism, which in general must mean normal, even if it's got functional differences, because millions of people around the world will have that form of DNA, and the rare variants that cause disease. So the other part of this, the idea that the site is the polymorphism that occurs in different forms, each of which is an allele, there's a great tendency to call an allele a polymorphism. It's the site that's the polymorphism. And also SNP, single nucleotide polymorphism, that you'll hear about more. So the types of polymorphisms are a combination of how one detects it and the nature of the variation. So the restriction fragment length polymorphisms was one of the first technologies. Those can be almost any of the other types. In terms of the DNA, it's basically a way of detecting variation, pretty much obsolete. The short tandem repeat polymorphisms, I always put the P on. In forensics, you generally hear of it as STRs, but it's the polymorphism part that's important. They're short tandem repeats. The VNTRs are generally longer segments that occur in tandem repeats, but similar in concept. Insertions, deletions, a bit of DNA from megabases, huge deletions in some areas that seem to be compatible with normality, down to 1 base pair, more or less. The single nucleotide polymorphisms, instead of an adenine or an adenosine, you've got guanosine, etc., just in the string of DNA. And then the copy number variation, where it's not a tandem repeat, but a segment of DNA may occur in 2 copies or 3 copies, sometimes tandem, sometimes a whole segment is missing, analogous to a deletion. So mitochondrial DNA. It's this small loop of DNA. It occurs in the mitochondrion. It's the remnant of an early parasite that invaded an early cell, and it's now dependent upon the nuclear genome, and we are dependent upon the genes here as the major energy-producing genes and apparatus of the cell. It has its own slightly different DNA code. It's got its own transfer RNAs for making proteins and its own machinery for making its own proteins, but most of the proteins are now made in the nucleus of the cell and imported into the mitochondrion. The relevant point here, of course, is that there are many polymorphisms. It's almost all coding So there are great restrictions on what variation can occur because it has to be compatible with function. So some variants are recurring. They have arisen independently many times because both alleles are compatible with functioning. The other thing is that the control region is highly variable because it only has limited function. As long as most of it's the same, the replication of the mitochondrion occurs normally. It doesn't code for a protein. So you'll see a lot of studies of hypervariable regions, which are highly polymorphic, and then of the single nucleotide or other variants around the rest of the mitochondrion. An advantage is that for every cell, there are 1, 2, maybe more thousands of copies of mitochondrial DNA. So it is much more prevalent in a sample than is nuclear DNA, and hence it has been studied because it doesn't need quite the sophistication and characterization as nuclear DNA. So a lot of human ancestry information and forensic identification has been based on mitochondrial DNA. Some of it is very powerful, but it's not as powerful as nuclear DNA. So basically, this summarizes what I've just been saying about the variation in both segments. But at any one site, there's not such a huge number of variants. Now, the relevant thing for ancestry and even individual identification is that the mitochondrial DNA is inherited only through the mother, because the little sperm is only a bundle of nuclear DNA that gets into the egg, but the egg comes already with all of the mother's mitochondria and hence her mitochondrial DNA. So even males have mitochondrial DNA, they just don't transmit it to their children. It's entirely based on what they inherited from their mother. So going back 5 generations, how many of your ancestors have your mitochondrial DNA? One out of 32, assuming there's no inbreeding along the line. My father was inbred because his mother and father were at least 5 generations removed from the 2 brothers who originally settled in the colonies, and they met just because the young soldier coming through town was asked if he'd met the widow kid and her 3 beautiful young daughters. So a young soldier, of course, wanted to meet them and married one of them. So within 5 generations, it is not that uncommon. So you're not learning much about your ancestry from this type of DNA. What about Y chromosome, the non-recombining part of your Y chromosome? Shows exactly the same pattern, except it's the paternal lineage. So again, you're not learning much about your ancestry, your overall ancestry, from just this. And I'll add that some of the companies that do ancestry testing will use only Y chromosome or only mitochondrial DNA. And they're not telling you a lot about your ancestry. So I mentioned the mitochondrial variation and the Y chromosome. There are relatively few genes, so there's a fair amount of single nucleotide polymorphism, and there are also many STRPs in the Y chromosome that have a higher mutation rate. Now, let's look at autosomal. Here is a sample pedigree where I've colored the alleles coming down. So we have ampersand and @ sign alleles in a sister, and we have number sign and star allele in a brother. They got the opposite alleles. Now, on this monitor, the green and the white don't show up very well, but what can be said about the ancestry who 5 generations ago contributed each of these alleles? Well, here we have the tracing back. And notice that this blue ampersand allele, as we go back, we have a homozygote and another homozygote. And so we cannot really identify where this particular allele came from. But it has to be one of those 5 ancestors. Similarly, the @ sign here, going back through the white, because here is white, it could only have come from here, even though both have the light green. That means the light green must have come from here. So from here, we can go back, again, a homozygote. It could have been from either of those or this ancestor. So we know roughly where that came from. We can go back with the hash mark, the number sign, and the star is the only one where we know that the father's father's mother's father was the origin of that particular allele. But clearly, overall, we're beginning to get a profile of the ancestry. And if we look at many individual loci, each locus is going to tell us something about some of those ancestors. And with many loci, because they're independent, we get a picture of all of them. So the next point is measuring variation. We know that these polymorphisms exist in frequencies, the individual alleles, in frequencies in a population. And a standard measure we use is called FST, for F as the letter we use for the inbreeding coefficient, subtotal to total, S&T. So in theory, it's related to random genetic drift. Has anybody heard of random genetic drift? A couple. You know that if you have 2 children, there's a 50% chance that you give to both of them the same allele that you have. And that chance over many people means that the gene frequency in a population of children will not be exactly the same as the gene frequency in the parents. And the smaller the number of parents and children, the greater the possible fluctuation would be. So among different populations, over time, random genetic drift causes some changes. And FST is a way, theoretically, of explaining that. So here's an example. I'm going to show many slides like this, and I always arrange African populations on the left, Mideast, European populations. Here's Western Siberia, East Asia, a couple of Pacific Island, Eastern Siberia, North America, South America. So here you see in black one of the two alleles. The two have a frequency summing to 1. So the other allele is 1 minus this, or coming from the top instead of the bottom. And you see two different polymorphic loci show different patterns of variation around the world. The expectation for any polymorphism you know nothing about in advance is that there's a lot of gene frequency variation around the world. We are all alike in ethical ways, but we are all different genetically. Even if we're from the same ethnic group, we're different. And so those differences become important. Here's another example, but these have low variation around the world. Same populations, same order. But they're not identical. One of the ways, just as an example, to look at genotypes, here are a bunch of individuals. Each dot is an individual. And this is using a TACMAN assay, which measures fluorescence as a function of the genotype. And so across the bottom, you've got the intensity of fluorescence for one fluor, and the y-axis, you've got the intensity of fluorescence for the other fluor. We type 384 individuals at a time. That's what each dot is. And so you can see the blue here represent individuals who have only the allele fluorescing in blue. They're homozygous. Only fluorescing in red, hence homozygous for the other allele, and a bunch of heterozygotes who fluoresce both colors. And the controls down here as black squares, and those samples that did not give an interpretable result. And here are some that we're not interpreting. Here's one, for whatever reason, low fluorescent, low intensity, whatever, not being interpreted. Here's one of the controls that's not where we'd like it to be. This is clearly real data, but it's certainly not up here. So it's not really affecting the interpretation. So that's sort of a little bit of background, but now exactly how are we using some of this DNA variation, the polymorphism, in forensics? So can be used to identify a criminal That's the way it's classically being used now. There's DNA from a crime scene. You've got the suspect's DNA. They match. And that's evidence for identity. Most of what we're interested in here is identifying human remains, or maybe from the crime scene trying to make some inference of the ethnicity or ancestry or the phenotype of the individual who left that DNA at the crime scene, on the supposition that's the criminal. But DNA is used all the time in parentage testing, and in the court system, the best use of DNA is to exonerate innocent people. I was once asked in cross-examination, if I were falsely accused of a crime and there was DNA, would I allow the DNA lab in the Royal Canadian Mounted Police forensic labs to test my DNA? And my response was, of course. It's the surest way I know to prove that I'm innocent. At which point the judge said to the cross-examining defense attorney, don't you think you should stop working for the prosecution and excuse this witness? So identifying human remains, you may have, based on what we experienced from the World Trade Center attacks, may have known DNA. Almost all of the firemen, the first responders, had given samples for bone marrow donation. And so there was a known DNA sample available to test. Clearly, a lot of relatives brought in toothbrushes, brought in dirty underwear, brought in all sorts of personal objects from which DNA could be obtained. And to date, I forgot to bring the number with me, but it's over 1,600 of the individuals have been identified with at least one little piece of bone. Determine the phenotypic characteristics. What hair color, natural hair color, did the person have? What skin color? Can we say anything about whether it was thick or thin hair? Can we say anything about height? Or determine the ancestry in terms of more indigenous, geographically indigenous origins. So the forensic question in matching to a known person is, first, what are the DNA patterns? So this is a molecular and a laboratory issue. Has the DNA been analyzed correctly? Have the patterns been interpreted correctly? Then, do the two patterns match? Is the method used specific enough that if the results are the same, you can say they match for that locus? Then the statistics. What are the chances that two unrelated people have the same pattern? Obviously, that becomes very critical, assuming the molecular is done well. And that's where databases are needed, because it all depends upon the allele frequency. If the frequency of an allele is 99%, and you've got 2 homozygotes for that allele, well, 81% of the population has both alleles the same. That really doesn't exclude a lot of people as not being the same, not very informative. And we'll get into that later. So the CODIS markers, the standard short tandem repeat polymorphisms used in cases nationwide now, are a panel of individual identification SNPs that are clearly appropriate for this kind of question. The lab methodology is pretty good, and they're fair databases. But individual identification isn't the only type. And remember, I mentioned earlier, the CODIS markers are not good for ancestry. They were picked because they are highly variable almost every place. And so there's not a lot of difference in allele frequencies among different populations. So I came up with a classification of 4 types a few years ago. There are individual identification SNPs. They have very low probabilities of 2 individuals having the same multi-site genotype. So each SNP is optimized, and the panel is very good. Ancestry informative SNPs would be sort of the opposite. The high probability that an individual's ancestry comes from one part of the world, or maybe admixed from 2 parts of the world. Lineage SNPs are where we're trying to get down to individual clans within a group, extended families, organized crime where it is a family. And the phenotype informative SNPs that will, based on allelic differences that control parts of the phenotype, will tell you something about how a person looks. So there are different requirements for these different purposes of using SNPs. And I'm concentrating on SNPs because that's really the best type of DNA for any of these applications in terms of laboratory methods, numbers of markers available, and the detailed annotation. So the importance here is that for the individual, the ancestry, and the clannish or lineage markers, they represent a small fraction of all available polymorphisms. So one wants to search for and optimize a set that is particularly appropriate for one of those purposes. The phenotype informative SNPs are also uncommon, but they deal with specific phenotypes. And as yet, though there are good candidates, they're poorly documented for exactly how they function in development of the phenotype. So there are now 5 or 6 loci that we know are clearly involved in the amount of melanin in the skin. But we don't know how they interact. So while we can type them and make predictions, their predictions are based on associations without a clear understanding of the interactions when you look at all of them. So, general criteria, I'm reiterating myself to an extent, readily typable as a unique marker, highly informative for the stated purpose, and well documented for such relevant characteristics as allele frequencies, association with phenotype, biology. So, which ones are going to be best? So we want the maximum amount of information per SNP, but what do we mean by information? And we want SNPs that are not subject to typing difficulties. And what kind of typing difficulties exist? So additional slides will amplify the first. Let me verbally amplify the second. Almost all of the typing methods involve using bits of DNA that are complementary to either conduct amplification of a fragment of DNA or specifically probe the small region around where the known variant is. But if there are other variants nearby that interfere with either of those, then one may not get an accurate reading. The test fails. And so if you've got a heterozygote, you only detect one of the alleles. It's not that the polymorphism is not valid, it's that that method does not detect it accurately. There are other problems that anybody working in a laboratory knows about: the phase of the moon, the what you ate for dinner the night before. All of these are probably real variables, in a humorous sense at least, but no method is perfect, no data set is error-free. We have to try to minimize them, and that's where the prior work will be best. So in terms of amount of information, we're talking about alleles, we're talking about allele frequencies, but what we see in the population is individuals who have two copies, one from the mother, one from the father. So fortunately, back in 1904, a geneticist asked a mathematician about this question, and Hardy-Weinberg came up with this very simple relationship based on elementary probability. And as a function of the gene frequencies, you can see here the genotype frequencies. And basically, p squared, 2pq, q squared is the Hardy-Weinberg ratios. It's the square of the quantity p plus q, the quantity squared, where p and q are the frequencies of the two different alleles. So it's very elementary probability. If we want the most diversity within a population, we clearly want the allele frequencies to be at 0.5, 0.5. So for individual identification, the lowest probability of somebody unrelated being the same is if the allele frequencies are equal to P equal Q equal 0.5. But remember I said they always differ among populations. So here's where the low FST, and here we're talking about heterozygosity. The frequency in this green line of an individual having two different alleles, being a heterozygote. In the zygote, they had two different alleles. So that's one aspect. For ancestry identification, we want the opposite. We want one population to be like this and the other population to be like that. So when we test it, we've got a distinction between the populations. So let's review now in terms of ancestry information a little bit about what we really do know about modern human evolution. This is, at its basics, no longer controversial, absolutely accepted. There's, of course, infinite argument about the nitty-picky fine details. That will always go on. This is science, and these are humans who are looking at it. But it's clear. Modern humans evolved in Africa roughly 200,000 years ago. And it's also very clear that considerable genetic variation accumulated in Africa, and it's still there. Where are the shortest people in the world? African pygmies. Where are, on average, the tallest people in the world? The Nilotics in Africa. And tremendous variation. In the US, among non-scientists, there tends to be, and even among some scientists who don't know much about human variation, there tends to be the assumption Africa is genetically homogeneous. Well, no. About 100,000 years ago, and here's where the argument, some say as recently as 80, some have even said 60,000 years ago, some individuals left Africa into Southwest Asia. And the single population had only a small fraction of the genetic variation present in Africa. And that population then expanded to occupy the rest of the world. And here is how I put it in a pointillist way. And this has been reproduced in National Geographic, and if you see the race exhibit that's going in museums around the country, it's currently at the Smithsonian in Washington.
This is part of the AAA online also, the race project.
Is it online? Well, it's animated in the museum. I don't know about online. But it's clear where just the different colors represent generalized genetic variation, that Africa had accumulated a lot, by 100,000 years ago. But notice, it's not uniformly distributed. There's a little more red here. There's a little more blue and yellow here, typical of any widespread mammalian or any other species. The fringes of the distribution don't have all of the variation. They're a little bit. That's gene flow and random genetic drift. Well, the last time I left Africa, it was in a 747 from Johannesburg. 100,000 years ago, the only way out of Africa was out of Northeast Africa into Southwest Asia. And we know that by 40,000 years ago, that population had spread. And if you look carefully, there is less variation out here than there is here, and yet it's dramatically less all over than it is in Africa. So basically, if you wiped everybody of non-African origin out, the human species would still have almost all of the genetic variation that it has today. Non-Africans represent a subset of genetic variation, and it's characterized by a loss of variation as humans have spread out of Africa, with a few exceptions, but they're the exceptions. So, random genetic drift can explain most of that, but selection also occurs. We all believe in evolution. That's selection. These things evolved to get food into mouth in part. I have no trouble using them to eat. How do we detect selection? We can argue that higher FSTs indicate selection in one part of the world than not. But that's hard. You have to be very specific, and it can occur by chance. So one of the methods of detecting selection is the idea that a particular variant in one part of the world has become common quickly, where random genetic drift to become common would take many generations. The result is less recombination in the DNA flanking it. So you tend to get around a variant that's been selected for an extended part of the DNA that is all identical. For example, what about lactose tolerance as an adult? Do you all know about that? Well, I've got the genes for it. Sorry, I just hit the mic. That is essentially fixed for one particular variant in Northern Europe. And it shows a climb, a gradient from low frequency in Southern Europe to higher frequency in Northern Europe. And very strong evidence of selection. The plausible hypothesis is that as the Neolithic moved north, your cow was in your hut with you during the winter when there was little to eat outside. And if you could use the cow's milk fresh, you would survive the winter better. And if you, as the hunter-gatherer during the winter, died, your children were going to die. So there's very strong survival value in being able to drink fresh milk. In Southern Europe, what happens to fresh milk? Converted to yogurt. So yogurt is a very part of that diet, but what makes yogurt? Lactobacillus that digests the lactose. So here we have culture and selection operating on a genetic trait. East Africa has adult lactose tolerance as well, but from a different independent mutation. There are many ways one can think of looking at selection, but they're mostly at the moment statistical until there's a solid biological explanation. And the one I just gave you is a good, story, probably makes sense, but it's not proof. So there are others. We know there are variants in hemoglobin. Everybody knows about sickle cell hemoglobin. There is proof. The different susceptibility of the different genotype to infection from the trypanosomes is clear. The survival of infants is clear in a malarial environment.